Hello and welcome to the second installment of my Fundamentals of Molecular Biology series. In this episode, we're going to continue where we left off, and we're going to be talking about DNA and especially why it's so important for molecular biology. So, section one, we'll talk about DNA as the genetic material, some of the experiments and rationalizations of why we need DNA as our genetic material, and even what genetic material is. We'll also talk about the structure of DNA and the packaging of DNA. You have 3 billion base pairs in your body. How do you fit that into one cell? And there's also DNA modification, which can occur and affect the readability of your genome. So, in section one, we have DNA as the genetic material. And when we think of a genome, we need to think about many parts working together to influence your phenotype or who you are. Because organisms contain some material that influences all of these characteristics if, we're naive and not already know that DNA is the genetic material, right? So all of life needs this genome, and we know from this genome, an idea of a genome, it has to have certain properties. So a genome abstractly should be replicable. It should be able to copy itself. So that's important. It should also be able to store information and act as a database for the cell. So you exactly is like a SQL database. You should have this sort of pure form of information the cell can go and read. So whenever this information needs accessed or queried, the cell can run over to this database, find the information it needs, and then use it or read it and access it. And it should also be variable, so information should also contain inherent variation across individuals. So, everything isn't a clone of each other, and you can have evolution and adaptation forms. So, this is what we're looking for in genetic material. I'll also include that, what's the big deal about a genome? We have NASA has recently found like DNA and RNA monomers on asteroids out in, I believe, the Kuiper Belt. And, presence of DNAs and RNAs on literal asteroids is sort of indicative of how life was able to form. Say it crashes on the Earth, and planets can be inoculated with DNAs. However, how you get from those building blocks of DNA to a fully functioning genome is something we don't understand yet. It's a gap in our understanding of how you can have this abiotic generation of life. So, talking more straightforward is that genetic information in our DNA can be transcribed into RNA and then translated into proteins. So that's what basically makes the meat. We're turning sequences of sugars into meat, basically, or proteins. And that in turn reflects a physical quality. For instance, your DNA says make arms, and then you have arms. So, in terminologically speaking, we have the genotype, which is an organism's inherited genetic code. So, everyone knows the genetic code like ATGC, whatever. The sequence of that and how it's read and how it's expressed and manipulated within the cell determines this phenotype, which is an observable set of traits in the individual. So, your DNA says brown hair, you're going to have brown hair. So, it's not necessarily that straightforward in reality, because if you change the genotype, of course you'll change the phenotype and result in a disease, but it doesn't have to be, you know, changing the actual bases or the actual code of your DNA. You can change how your code is read, for instance. So if you change the usability of your genotype, you're going to change the phenotype, and that is epigenetics, and that's a whole other domain. So now this is sort of the history of how we determine that DNA was the genetic material. So Gregor Mendel in the 1860s, so the Civil War era, showed that traits were not inherited directly, but were specified by genes passed down from parents to offsprings. This is where we get the Punnett squares from simple Mendelian genetics, like homozygous, heterozygous. So everyone's had some biological background, but you know what a heterozygote is, what a homozygote is, likely has molecular underpinnings at the DNA level. So in the 1860s, we didn't actually know what this material was that was making these heterozygotes, homozygotes, whatever, and what even was responsible for this. We just knew that you could predict the inheritability of traits. So some time passes until the 1920s, when Frederick Griffith was trying to develop a vaccine for pneumonia, discovered a transforming principle which allowed non-deadly R strain bacteria to take up some genetic material from a deadly S strain heat-killed bacteria. So basically you have this two strains of bacteria. One is rough strained. I guess you could see that it physically looked rough, and one was more smooth and was virulent and would actually kill the mouse. So if you have a heat-killed smooth strain, of course it doesn't do anything. And if you have a heat-killed or regular rough strain, it doesn't do anything. But if you take some material from the smooth strain and put it into the non-virulent rough strain, then it can kill the mouse. So, okay, you can transfer some information from one thing to another. So it took us until the 1940s to advance again. You can see these gaps. Biology is kind of a emerging field. We really don't know a lot yet. It's only been 80 years of like hardcore biological research. So Avery, McCarthy, and MacLeod wanted to identify the material responsible for this transforming principle. So they're going to use enzymes to selectively destroy either proteins, RNAs, or DNAs, where only DNA digestion in their experiment prevented the transforming principle. So there was still some uncertainty after this regarding the purity of the batch, but in contemporary analysis, this is the first experiment that really shows that, hey, if you remove the DNA, you're not able to make these transformed R cells that are able to gain the killing function. So it's not RNA and it's not protein. It's only the DNA that can transform these cells. So again, DNA was the heredity material that phages would use to infect cells, and it carries the readable information. So that brings us to, although scientists knew that DNA was the hereditary material then from that experiment, nobody knew what the actual structure was. So in the 1940s, again, Aaron Chargaff identified that in DNA from several organisms, the composition of adenine, or A, is proportional to thymine, and guanine was proportional to cytosine. So if they're one-to-one, they could be interacting. So then in 1953, Watson and Crick discovered the double helix structure of DNA utilizing those Chargraph rules and also some chemical understandings, both from the basic chemistry field and also from Rosalind Franklin's X-ray crystallography. Now these guys are kind of contentious in the field. I believe Watson was actually in the Epstein files, but um, the the actual discovery is that we know from them that DNA is this double helix structure composed of nucleotides. So there's four nitrogenous bases affixed onto deoxyribose sugar along with a phosphate group to create a nucleotide. So what does that mean? Oh great, you can see my little red dot. So we have adenine, guanine, thymine, and cytosine. So you have your purines here, which have sort of these two rings on them, while pyrimidines have this one ring structure with a lot of nitrogen or whatever. And what your cells are able to do is form these nucleotides from these nitrogenous bases, which is basically throwing a sugar backbone of ribose on here and a little phosphate group. So the actual composition of the genetic material is, I believe, selected by DNA with intention. So the sugar molecule is very important, and two prime ones exist in the molecular biology landscape that you should pay attention to. One is ribose, which is this pentose sugar here, kind of like fructose, but with some extra flavors on it, and deoxyribose. So the D in DNA, and that gives away the story of what the actual sugar is. But you can see on this second position here, so you read in organic chemistry, you read carbons in a linear fashion, so 1, 2, 3, 4, and 5. So the second carbon in deoxyribose doesn't have a hydroxyl, it's just hydrogen. And if you read more into organic chemistry, you sort of need reactive groups like oxygen or nitrogen or more electronegative functional groups to do chemistry. Hydrogen doesn't really have it. It's just a proton and then an electron circling it, so it doesn't have a reactive potential. So the only part with reactive potential is the third carbon, which becomes quite critical, where you can form phosphodiester bonds between these nucleotides by reacting that 3' hydroxyl group with another nucleotide's 5' phosphate. And chemically you're able to link these then together of this 3' through the 5' phosphate group. And this creates a directionality because you can only extend your chain in one direction. So DNA can only grow in a 5' to 3' direction. Again, 5' is referring to that phosphate and the 3' to that hydroxyl. So, you're able to extend one linear chain this way. However, in the cell, DNA polymerase will facilitate this reaction. You don't have to worry about like the synthetic chemistry of how it's all possible or whatever. Just know that it requires the 3' hydroxyl to exist and be able to be built off of. So, more on the structure of DNA. From Chargraph, we know, as I talked about previously, that a purine must dissociate with a pyrimidine because the proportions were similar. And electrostatic interactions will also form between these molecules, like hydrogen bonding between these hydroxyl groups and nitrogens, which will allow enzymatic manipulation of these strands. So as you can see from the A and T hydrogen bond interactions and the guanine and cytosine interactions, there's only two hydrogen bonds between the adenine and thymine based on their chemistry. However, with the guanine and cytosine, there's three. So if you think about this on sort of a macro level, the A and T's are going to be weaker than the C and G bonds because there's less hydrogen bonds. And this becomes important sort of in telomere regions, or I suppose we'll discuss later in this lecture. So also, as I talked about previously, DNA is directional because it can only build it in one direction because of how that chemistry is. So the only way for these interactions to occur, so to get A and T and G and C, is to have these strands grow anti-parallel, yet be complementary. So you have a 5' to 3' this way and a 5' to 3' this way, yet they're have complementary bases, but the chemistry is going opposite ways. See, the hydroxyl is down here, but the phosphate is up here. So that's what is the essence of anti-parallel strands. So in the 3D structure of DNA, there's also major and minor grooves which form in the DNA double helix because the bases are not attached symmetrically to the sugar phosphate backbone. So there's a consistent amount of wiggle or give, whatever the chemical understanding is. And this is functionally significant. So major grooves will open up in the DNA structure because of this asymmetric binding, and sort of will expose the chemical information within your DNA so you're able to read it and access it. So transcription factors will be able to recognize like what base is in there, and this is your search function, right? If the DNA is your database, your transcription factor will control F and kind of look for the bases in your DNA on the major grooves. It doesn't look on the minor grooves because they're harder to read. You can see they're kind of smashed together and more compressed. So an example of this is actually the Mic-Max transcription factor, and it's a heterodimer, meaning that this Mic protein is produced by one gene and the Max protein is produced by another gene, and when they fuse together, then they form the working transcription factor, which is like a pair of scissors, and it'll use a helix loop helix leucine zipper motif to try to find the major groove on DNA. And it's sort of specific for EBOX sequences, which is CAC-GTG, primarily through the major groove. So now we're going to pivot into DNA packaging. So the human genome is massive, meaning there's 3 billion base pairs in every single one of your cells, and there's trillions of cells, so the amount of information in one person is incredible. It's more numerous than like the stars in the Milky Way. So how you organize the genetic material and how do you compact it into the cell so it's even readable or workable and organized. So chromosomes consist of actually DNA and packaging proteins that will help the DNA condense into a way smaller footprint so that it's workable and transportable. So the actual DNA protein complex is chromatin, and there's some confusion between what chromatin is and chromosomes is, and that's because biologists are really great at naming things, and I hope I'll be able to elucidate it when it comes up. But DNA condensation will help protect the genome from extra genomic things that can hurt your DNA or mutate it, but it can also affect its readability, right? If you compress it, how are you going to stick your transcription factor in there? Like your major groove won't be accessible. So the main packing protein in the DNA is called a nucleosome, and we're going to discuss how that works. So nucleosome protein is called a histone. So histones are the main proteins that DNA wraps around to condense, and it's actually an octamer composed of four core monomers: H2A, H2B, H3, and H4. And you can see they're like pie wedges, and they'll come together and form this octamer protein. Now, chemically, histones are positively charged with a lot of lysine and arginine residues, so they put out a lot of positive charge. However, your DNA, because it has a phosphate group and a lot of oxygen, which is negatively charged, um, they'll naturally attract to each other through electrostatic interactions, right? Opposites attract. So that's sort of the evolutionary reason why you have these positively charged histones wrapping around your DNA and sort of helping to condense it. So DNA will be, again, wound around the octamer in this left-handed direction. So when the DNA is unwound, it can induce negative tension and help it dissociate or open up more. So histones also have some tails with which can interact with other nucleosomes and also interact with a helper protein H1 to help compact the DNA even further. It's all about really squishing it down and making it way more manageable for your cell to use. So once you add some octamer to like a string of DNA, you'll get a 10 nanometer fiber that will arise, and then it will further compact and lead to a 30 nanometer fiber. So the tails can also be chemically modified to affect the chromatin structure, but we'll talk about that later. So whenever you're compressing the DNA, you need to think about, what is that doing to the readability of your DNA? So you can create something called heterochromatin, which will be transcriptionally inactive chromatin from really dense histone packing, so you're not going to be able to throw your transcription factor in there and read what the actual DNA is. And there's two sort of types I'm going to talk about in this lecture, which is constitutive heterochromatin, which is always condensed and packed tightly because you don't really need it. It's not really meant to be read. It's more serving a functional aspect to protect your genome, such as telomeres, which are at the end of DNA because DNA is actually in human cells is linear, so you can imagine a very long string with ends. Now, what can happen at the ends if you don't have something clamping down like two strings together is it's going to unravel and you're going to have a nightmare in your genome. So the telomeres and a bunch of protein at the end helps clamp it down so it doesn't unravel and shear. And there's also facultative chromatin, heterochromatin, which will switch between highly and lowly condensed. So this is sort of modular. An example of this would be during developmental biology. Some genes will be active and help like differentiate your cells from like embryonic stem cells into like muscle cells or liver cells or whatever. So you don't need everything firing at once. You can kind of control that with various factors from the mother. So there's also euchromatin, which is transcriptionally active chromatin with less histone packing density. And during metaphase, chromosomes will form identical sister chromatids connected at a centromere. So this is sort of a confusing topic for biology students, but chromosomes are identified by the number of centromeres in the body. So your chromosome count doesn't change as the cell progresses through the cell cycle where there's DNA replication. So in humans, our chromosomes will always be 46 chromosomes. That number never changes because chromosomes are again defined by the centromere, and the centromere count never changes. So the centromere contains a lot of nucleosomes, and it's sort of this core of the chromosome. It has a lot of H3 and the CENP-A protein, and that whole complex of the protein and the DNA and the location at the centromere is called the kinetochore. Again, with the telomeres, at the end of your DNA, you have TG repeats at the end of the chromosomes, and it will help prevent your DNA from fraying, help it from sticking to other chromosomes. And also at the end here, we'll talk about later in the DNA replication lecture we'll have, is the loss of genetic information. So eventually you're going to pack your DNA so tightly it becomes a chromosome. It's already a chromosome. What I mean by this is you see it become the X shape you're familiar with whenever you think of a chromosome. So this will protect the DNA during segregation, during the cell cycle. So again, we'll talk about that more in depth when we have a cell cycle lecture. And again, humans have 46 chromosomes, and 23 are pairs of homologous or duplicated gene. Well, not necessarily duplicated, because you can have different alleles, and this is where you can have heterozygotes or homozygotes from, but everything has a molecular basis at this point. So when the DNA is compacted this tightly, it cannot be read. So this is really a transient form of DNA, really for facilitating the cell cycle. So the nucleus is the storage site of DNA, and the relative positioning of DNA in the nucleus is actually consequential. So chromosomes are maintained in the nucleus in discrete territories called chromosome territories. Very creative naming there. Where transcribing regions are also ordered between inactive domains, A-B compartments, which will also help perform specific transcriptional functions. So again, when the cell is not replicating, DNA exists in more of a relaxed euchromatin or a condensed chromatin state, and the cell will kind of put its euchromatin more towards the center and allow transcription to occur there, and it will put the heterochromatin out towards the periphery. So also, A compartments are these gene-rich and actively transcribed regions. And these regions are located, again, at the interior of the nucleus, because that's where all the euchromatin is, and they'll interact with other active chromatin regions, whereas the B compartments are gene poor and transcriptionally inactive, and they'll be towards the periphery of your nucleus and interact with other inactive chromatin regions. So you can see on my channel I actually have a dedicated video to AB compartments and the organization of the nucleus, so you can watch that if you want more. So what you need to know is that A and B compartments are not exactly euchromatin or heterochromatin. AB compartments are sort of this big picture, while euchromatin and heterochromatin will describe the accessibility of the actual local state. So on a much smaller scale, and is a dynamic property of the DNA. So chromatin remodeling enzymes will also use ATP hydrolysis to move, eject, and exchange these histones. And histone modification enzymes can attach chemical groups to these histones and help recruit other proteins. So because these features are actually not hard-coded in a DNA sequence, but they have this impact on how DNA can be accessed and read, that's the essentiality of epigenetics. So changing the readability is going to change your phenotype. So more on that we have DNA modification, where you have histone acetyltransferases, which will throw acetyl groups onto those lysines in your histones, and you're masking the positive charge. So reducing that electrostatic interaction between the positively charged histones and your negatively charged DNA, it's going to open up your chromatin, relax it, turn it in that euchromatin, and allow transcription. And the reverse is your histone deacetylase, which is going to rip off the acetyl groups on the lysine, expose the positive charge, allow interaction between the negatively charged DNA backbone and the positively charged histone to function again and close your chromatin and reduce expression. So that's a main how euchromatin and heterochromatin transition can occur with these two enzymes. And there's also some tagging which can occur and is more nebulous in function, where you have histone methyltransferase, which will add methyl groups onto lysine, and you also have histone demethyltransferase, which will remove the methyl from your lysine. And methylation, again, will mark activation such as H3K-ME3 at your active promoters, and it will mark repression at other things, so it's more nebulous. And you read this as, there's a special way to read this, so this is H3, so the histone protein H3. K4 is saying the fourth lysine has 3 methylations. So they don't change DNA's charge, it'll act as a signal to recruit regulatory proteins. And this will also help mark DNA damage, which we'll talk about again in a subsequent lecture, so H2AX will form. There's also various anti-cancer drugs which are important that exist to target these enzymes for therapeutic effects. Many are HDAC inhibitors, so varinostat, romdepicin, belinostat, panobinostat, I believe are FDA approved. So varinostat will directly bind to the catalytic pocket of HDAC enzymes, and it's orally available and used to treat T-cell lymphoma. But blocking HDAC is thought to actually prevent tumor suppressor gene silencing and allow histone acetyltransferases to get in there and reactivate genes which were epigenetically silenced. So whenever you encounter these drugs or these systems, just think about the actual logic of it, like what happens, and you can reason the mechanism from that. So in a similar vein, instead of modifying the histones, you can modify the actual DNA. So DNA methyltransferases can add methyl groups on the cytosine and make 5-methylcytosine. And this is used in eukaryotes such as humans to silence transcription. So leading up to most of our genes in DNA, you'll have things called CPG islands, where you read it as cytosine and this is the phosphate between them, and of course guanine. They'll be methylated, and 60% of human CPGs are methylated and actually inheritable, so you get this from your parents and you can transfer it on to your progeny. But DNA methylation changes in tumors, so cancer will show that hypomethylation, so ripping off a lot of those methyls and just leaving like pure DNA, can lead to gene expression upregulation and contribute to genomic instability. So actually it's using these methyl groups to silence transcription and prevent transcription factor binding, because you're changing sort of the electrostatic interaction of the transcription factor and the DNA, right? If you throw something on there that's preventing the proper binding, it's perturbing that interaction. So DNA methylation also will silence gene expression. Now another way is actually the topology of DNA. So you can manipulate the DNA topology and sort of induce stress in your double helix. So you can open and close the DNA during processes we'll talk about in subsequent lectures, such as DNA replication. But every time you open it in sort of this bubble shape, it'll cause torsional strain in positive and negative directions. So upstream of your bubble and downstream of your bubble, it'll cause like overwinding. For instance, if you had a string and you sort of try to open it up, part of it will become overwound and too tight, and part of it will become underwound. So, in order to fix this in your DNA, because that could be catastrophic and cause a lot of stress, is we have topoisomerase enzymes, which will sort of go in there and relieve this strain, so everything's nice and topologically sound. You're not inducing a lot of stress. And there's two types we have, where a type 1 enzyme does not require ATP, and it will make a single strand break and relieve tension and religate it. So, this is sort of a efficient way. You're not using ATP, and you're able to relieve your strain. However, more prevalent are the type 2, which will require ATP, and it just cuts both strands of DNA and allows the strain to sort of spin out, and it'll glue it back together. And both are active at the sites of DNA replication, but type 2 is sort of more important pharmacologically speaking, where we've developed drugs like doxorubicin, which is actually an anthracycline drug, which maybe we'll talk about more in the future. But it will stabilize DNA top 2 complexes after strand cleavage and actually prevent the religation of the complex causing cell death. So the thing you need to appreciate about the actual anti-cancer pharmacology from this slide is that, we can drug it and, prevent sort of these interactions from forming and resolving or whatever, but doxorubicin has really high toxicity. You can imagine this affects every cell in your body, but it's also really effective at killing cells. So although it's a major frontline agent for treatment of many cancers, it's also pretty toxic, and you're hurting a lot of perfectly fine cells. So that's all I have today in terms of content. I hope you enjoyed this lecture and were able to take something away from this. In the next lecture, we're going to talk more so about the actual mechanism of DNA repair, its role in the cell cycle, and DNA damage and repair, which can also occur. Thanks for watching.